Hi, I'm Joe Liu. I'm from UCLA, and I'm happy to be the moderator of this Proteoform Thursday meeting. Let me just first turn it over to our fearless leader for the Consortium for Top-Down Proteomics. That's Paul Danis.
Great, great. Thanks, Joe. Welcome, everyone. It's great to have you join us on this morning, afternoon, or evening, depending where you are. A couple things just to mention about upcoming events and some of the news. But first, we'd like to thank our annual sponsors who provided the support we need to do these kinds of events. Thermo Fisher, Bruker, SciEx, and Lilly are our major sponsors, as well as our supporters, Agilent, Pfizer, Nautilus, Biotech, New Objective, and Impro. We thank them, and if you see them, you can thank them as well. And additionally, if someone else would like to participate in the community and support these activities, just let us know. We'd be happy to speak to you about some of our offerings for sponsorship. Just a quick notice, some of you may not have seen or may not be familiar with on our website, we have some method papers there, a variety of methods. If you knew or if you wanted some more input on different approaches you can take, we have two that are particularly useful. One is around separation methods in mass analysis methods for intact proteins. A lot of people, when they move from peptide world to protein world, one of the biggest challenges is how do you separate proteins? It's a different universe, and we have a very nice paper here. It's been cited over 300 times, and the details are all in the paper. It's on the website. You can see that there. Additionally, we have a paper around native MS and how you can approach this, some of the basic guidance around methodologies you want to try there. So that's also online. You can take a look at that as well. Coming up, a lot of you are familiar with the ASMS meeting. We have the top-down proteomics short course. There's 2 days of it. One day is a lot of the basics of how you do top-down proteomics. The second day is focused just on the data analysis and visualization side of it. So that's online. I think that isn't there a deadline, April 30th or something, a deadline for this, Joe? Or
Yeah, I think so. Yes.
yeah, so take a look at that if you're interested, or if you have colleagues who are moving into the field that want some really good information from some of the leaders in the field. You can go there and take a look at that. As far as upcoming events, in May we have Boris Kriekel from Hamburg, and in June we have Mohammed Zanaidi from APAF in Australia, and in August we have Ana Marinho from Oswald Cruz in Brazil. So exciting talks coming up from around the world from some of the early career researchers doing some very nice work. So with that, I'll turn it over to you, Joe. Thank you up for everyone, and we look forward to hearing Emily's story.
Thanks, Paul. So it's my pleasure to introduce Dr. Emily Byrd from University of Leeds. Um, if you don't know Emily, let me just give you a little bit about her background. She received her bachelor's degree in biochemistry from Sheffield Hallam University in the UK. Then she moved 37 miles north to Leeds University, where she received her PhD in molecular and cellular biology from University of Leeds, working with Frank Sobat and Sheena Radford. And while she was working with Frank and Sheena, she worked on the structural dynamics of this protein α-synuclein involved in Parkinson's disease and did a lot of structural mass spectrometry work, including eye mobility, HD exchange, native mass spectrometry, to study how metals then might induce compaction of these kinds of intrinsically disordered proteins as they go on to amyloid formation. She then moved, oh God, maybe 100 feet. I'm not sure. I've never been well, I've only been to Leeds once. So she then moved about 100 feet then for a postdoctoral fellowship with Anton Calabrese. So she's still at Leeds. And there she picked up this unique method of using nanopipette electrospray ionization, which she'll tell you a little bit more during the talk, for native mass spectrometry to probe the conformational landscape of other intrinsically disordered proteins, such as TDP43, that's found also in Alzheimer's diseases in the presence of biochemically relevant buffer systems. So during her postdoctoral fellowship, then she did actually move a little bit more than 100 feet. She did a 3.5 month visiting research position at my lab in UCLA. So she crossed across the ocean, across the country, came to UCLA and worked with my laboratory to see how nanopipette electrospray ionization works in our systems. She was supported by a British Mass Spectrometry Society Research Fellowship for that stay. And now she's back in Leeds now. And so while she's been in Leeds, then she's published at least 8 publications that I remembered. Five as first co-author. And she's received many awards, including the most recent award. She's received a Marie Curie Global Fellowship Award. And that's a big award. It's a fellowship that allows her to come back to UCLA for a couple of years, and then also then go to the Free University Amsterdam in the Netherlands then to work with not only myself as ASM as president, but also with Anouk Ries, the president of the Dutch Society of Mass Spectrometry. So she gets to work with 2 presidents in her 3-year fellowship. So I will stop talking. I will let Emily then tell her about our research, about her research, not my research, where she's going to tell you about how nanopipette electrospray ionization can enhance your ability to do native mass spectrometry and top-down mass spectrometry. So Emily, take it away.
Cool. Thanks, Joe. Thank you for calculating my distances traveled.
Which is not much, really.
No, it was like very small, and then it's going to be very big.
Yes, that's right.
Lovely. Okay, can you see my screen okay? Is it all good?
Yes, yes.
Okay, so yeah, as Joe mentioned, what I'm going to talk about is some work that I started at the University of Leeds during my postdoc, and then continued during a research support grant funded by the British Mass Spectrometry Society in Joe's lab. And this is work on using what we call nanopipettes for native mass spectrometry. So I don't need to introduce electrospray ionization here, but a point I will make is that electrospray ionization can inform on protein conformations as a sort of snapshot from what they were in solution before we transferred them to the gas phase. So if we have a really folded protein or one with a 3-dimensional structure, such as lysozyme here, lots of charge sites are sequestered or protonatable sites. So you generally get these very narrow charge state distributions. Whereas if we look at a protein that's intrinsically disordered, such as α-synuclein that's involved in Parkinson's disease, as Joe mentioned, we get these really broad charge state distributions. So this protein's an IDP, it lacks any 3-dimensional structure, so it's really exposed to solution, so it can pick up lots of protons during the ESI process. And you get these really high charge states on the left over here, which are from these really expanded conformations. But one important thing that we know about intrinsically disordered proteins is that they transiently occupy these partially compact conformations that are stabilized by transient intramolecular interactions. So we also get this abundance of lower charge state species here that represent those partially compact species. So in terms of electrospray ionization emitters, typically ubiquitously across the field, we'll use borosilicate capillaries that have a pore opening or an internal diameter of around 2 micrometers. And there's been some really nice work from a few labs, particularly Evan Williams in Berkeley, where they've been looking at manipulating the protocols for making emitters to make them smaller and whether that can allow us to spray proteins in different buffers. Because as we know, as native mass spectrometrists, we have to either buffer exchange our proteins into ammonium acetate because it's volatile to enable them to go into their gaseous phase. So if we, in this paper, they had a BSA, ConA, and ADH, and if they tried to spray them in a salty buffer, so here they've tried KCl with tris, you don't really get a resolvable charge state distribution, you just get this broad hump and lots of salt clusters in the lower M/Z range. Whereas when they manipulated their filament puller protocol, still with borosilicate emitters, to generate tips that have a internal diameter of around 500 nanometers. So these are going to generate much smaller droplets, and this reduces non-specific adduction. We can see they've got some really nice spectra where they're able to resolve charge states for monomers of BSA and also oligomeric states such as dimers and tetramers for ConA and ADH. So there are alternative approaches for desalting proteins and biomolecules for native mass spec. So one of those is using a similar approach, so dual barrel feeder emitters, and these feature like a split capillary in the tip, so you can perform incomplete mixing with volatile buffers such as ammonium acetate and generate really nice resolvable spectrum and clean them up. There's also other techniques such as this instrument from Anson Biotech, which is called the Dynachip, and this relies on microfluidics, so really low volume, rapid buffer exchange. And they're bringing out some really nice examples. They're bringing more and more examples out all the time that show that the Dynachip can clean up biomolecules prior to native mass spec. And I believe this source can roll up to most instrument vendors. And then there's also online buffer exchange, which relies on chromatographic separation. So, for example, positioning a size exclusion chromatography column prior to your mass spec inlet for rapid buffer exchange. However, each of these have some limitations to them. So, for example, online buffer exchange, your mass spec acquisition is very limited. It relies on these very short elution profiles from your chromatography system. So you can't really acquire over long periods of time. So our approach is pretty simple. We use what we call nanopipette emitters. Firstly, I'll just explain again the standard setup so we can see the differences between standard emitters and our nanopipettes. So standard emitters are borosilicate glass. We pull them using a heated filament puller, and then we pull them closed, so you clip them open using metal tweezers. And of course, this procedure is done by eye by different scientists, so it's kind of not a little bit non-reproducible. But you also coat these tips in metal, such as palladium, so that when you apply voltage to the tip, it essentially becomes an electrode, and the protein spray or biomolecule spray from it becomes charged. Or alternatively, you can insert platinum wire in the back to charge the solution rather than the tip itself. So when we pull these and then clip them to size using tweezers or however you do that, we get tip openings in the micrometer scale, so usually around 2 micrometers. Whereas at Leeds, we collaborated with colleagues. Oops, sorry. Bear with. I've just lost my screen. Sorry. How do I get it back?
That's funny. We can see it here.
There. Oh, I got it. Sorry. I might have to stop sharing and reshare. I'm so sorry. It's because Joe, your face was like over half the screen, so had tried to move you. Okay, sorry, I'm back. Can you see it okay? Is it back?
Yes.
Okay, thank you, sorry. At Leeds, we collaborated with colleagues in the School of Engineering, Professor Paolo Actis and Dr. Charles Chow, because they saw how we make our ESI emitters, and they thought, well, we make nanopore electrodes pretty similarly in a similar fashion. The differences are that they use quartz glass to make their nanopores for things like electrochemistry and nanopore sequencing, because quartz glass has a higher conductivity, so you can use much higher temperatures to pull emitters. So you can use a Sutter P2000 laser puller. And one thing we know about pulling emitters is that temperature is a big factor for determining pore diameter. So with quartz, you can use much higher temperatures, so you can make much finer pores. So yes, we use quartz glass and a laser puller, and we generate 2 ASI needles or nanopipettes. We don't coat these in metal because we're worried about blocking the pore because it's so fine. Instead, as I said, we put platinum wire in the back and charge the solution rather than the tip itself. And because these tips have a much finer diameter, here an example from an SEM image, they're around 30 nanometers, we get a much finer droplet spray coming out of them. Oh my goodness, I'm so sorry. I don't know what I've done. Okay, thanks. So yeah, just briefly go back over that. Standard tips, borosilicate glass, and then they're pulled closed, and then you clip them to size using tweezers, and they end up in the micrometer range. Whereas nanopipettes are quartz glass. We use a carbon dioxide laser from a Sutter P2000, and we generate tips in the nanometer range. So some examples of how we've applied nanopipettes. These are quite specific at first. As Joe mentioned, I worked a lot with α-synuclein in my PhD and postdoc. So what we wanted to do was see if we could start to spray proteins in different buffers or biochemical buffers rather than just ammonium acetate, because we can make use of this finer droplet spray. So on the left is an example of a clipped standard capillary, and you can see like this process is very non-reproducible. The tip size here is around 16 micrometers. And if I tried to spray α-synuclein in PBS buffer, what we see, as you'd expect, is a mess. It's highly adducted, so the signal is spread over all of these adduct peaks. We have this rising baseline, and also the abundance of salt clusters in the lower M/Z range. Whereas if we then switch to using nanopipettes, here at Leeds, we make them around 30 to 40 nanometers in diameter. You can see that we massively clean up this spectrum. We get a resolvable charge state distribution, less adducts, and less of these salt clusters in the lower M/Z range. And we did this on a Synapt G1. So the nice thing about that is that we can couple our native mass spec with ion mobility measurements. And in doing so, in the amyloid field, one thing that we've seen is that different labs do amyloid kinetics, so the time taken for α-synuclein to go on to form amyloid fibrils, in all sorts of different buffers, and then all labs tend to see different rates of amyloid assembly. So one thing that we thought is that the protein itself, even though it's an IDP, it might be conformationally different in space in different buffers, and that might be why it goes on to form amyloid at different rates. So we were able to test α-synuclein, sort of get a visual of the conformational fingerprint by ion mobility in different buffers, so in ammonium acetate here, compared with PBS, because we use nanopipettes. And what we can see is that the conformational landscape is different. So the average CCS moves between these buffers, and the population of compact versus expanded species changes. And because this is a top-down seminar, I don't have a great deal of top-down data, but I thought I'd chuck this in because one other thing we tried to do with this was to see if we could use top-down fragmentation as a way of also probing conformation. So whether this IDP, when it forms these partially compact conformations, whether we can see differences in fragmentation efficiency for different local regions of the protein, and if we see lower efficiency, it could be because it's forming these partially compact regions locally. The sequence coverage isn't great because I used electron-based fragmentation, and the C-terminal domain around here of α-synuclein features lots of negative residues, so lots of Ds and Es, and of course that lowers our efficiency of electron-based fragmentation. But what we can see is that the fragmentation patterns are generally kind of similar between these two buffers. So here I have ammonium acetate and then also 20 millimolar tris that I've sprayed with nanopipettes. You can see there's minimal adduction from the tris. And the reason that they might be quite similar is one, because they use the same charge state, but also because the actual window of CCS values is kind of similar between these buffers. It's just the population of compact versus expanded species that changes. And then really quickly, one other example of the way we've applied nanopipettes here is another intrinsically disordered protein that's involved in Alzheimer's and ALS called TDP43. So here I'm just working with an isolation of the C-terminal domain of the protein, which is on the right. It's predominantly intrinsically disordered, but it also features this small alpha helix, which is transient within it. And some other groups have shown that in different ionic strength and in the presence of salts, the sort of biological function and dysfunction of this protein can change. So we wanted to see if we could visualize that using native mass spec and look at the conformational changes. So on the left is native mass spectrum with no salt, and then with the nanopipettes, we're able to spray things in 150 millimolar sodium chloride, which usually no one would want to spray by a native mass spec. And we can see that the charge states move, so we lose these lowest charge states, so suggesting that the protein is becoming more expanded. And then we can match that up with our ion mobility measurements and see that the average CCS does indeed move and get more expanded with in the presence of salt, so in higher ionic strength. And then we go on to talk about how this, we did a lot of biochemistry and computational methods as well to show that this expansion alters the pathway that this protein takes to form either amyloid or undergo a process called liquid-liquid phase separation. However, these two examples that I've shown you, they're very specific, they're very niche. So they're both amyloid proteins, they're both very small, they're 14 kilodaltons, and they're both intrinsically disordered. So then I got to Joe's lab and we were like, let's see how far we can expand our amenable analytes to nanopipettes to see what kind of molecular weights and what kind of systems we can spray from these tips rather than just focus on these small intrinsically disordered proteins. Oh, bear with. I will talk about that in a second. I have one more example that I forgot about. Um, so one more example, sorry, of this protein, TDP43, is that we tried to use fast photochemical oxidation of proteins, which is a covalent labeling method, as a way of probing protein solvent exposure. So TDP43, I mentioned, it's disordered, but it's also called a low complexity domain. So that means the sequence is really boring. There's no charge residues. It's pretty much just glycine, so it's not very fun. And that means when we perform FPOP, and the way that FPOP works is you have your protein mixed with hydrogen peroxide. You then excite hydrogen peroxide and break it up into hydroxyl radicals using a excimer laser at 248 nanometers. These radicals then rapidly, on a microsecond to millisecond timescale, attach onto solvent-exposed residues in the protein. And there's also a hierarchy of amino acids that will react with radicals. And then you quench the reaction to remove any free hydrogen peroxide so you don't overreact the system. And then you'll digest it, typically using trypsin or other enzymes into peptides and use MSMS to track where these modifications have occurred. However, with this protein that I've been looking at, whilst we want to probe these solvent accessible regions to see if these compactions or expansions are occurring, it's a low complexity domain. So digesting it using trypsin or gluc isn't going to look very good because our coverage is going to be really poor. So we're not going to get a very good read on local modifications. So I thought maybe we could try and do this natively top down now that we use nanopipettes, because FPOP is typically done in like a phosphate buffer. You have other things present, you have scavengers, histone and methionine. There's all sorts of messy things in there that would make this really difficult to look at natively. So here is a spectrum, native mass spectrum before FPOP, and then really nice when we spray with nanopipettes, we get this nice clean spectrum post FPOP, and you can see that we can see these hydroxyl modifications, these oxidations to the charge states. And then you can zoom in on those and isolate singly or doubly modified peaks to try and localize where these modifications are occurring. And I said I'm going to chuck a little bit more top down in. So this was done with Ken Durbin proteinaceous using ProCyte Native. And just really quickly, we can start to localize where modifications are occurring. So if we take a 2 times modified or a singly modified peak, we're localizing these to methionines that are in disordered regions, which is really nice because methionine is very reactive with hydroxyl radicals. Okay, but yeah, as I said, these are very specific systems, and we wanted to see how we could broaden our applications of nanopipettes. So first thing we did was we just took some standard proteins that are very small, so myoglobin around 17 kilodaltons and carbonic anhydrase, which is around 29. And all I did was just dissolve the lyophilized protein, but I didn't perform any buffer exchange. I just sprayed them straight away. So with standard emitters, you can see that they're quite salty. They're very adducted in both cases. Whereas when we switch to nanopipettes with no buffer exchange, you can see they're already really effective at cleaning up the spectrum, reducing the number of adducts in both cases. But it looks so much better when you zoom in on the most intense charge states. So myoglobin here with standard emitters, you can see all of these sodium adducts. And then when we spray with nanopipettes, not performing any laborious buffer exchange or anything, you can see these adducts have been reduced. And then again with carbonic anhydrase, when you zoom in, you've got these sodium adducts here. And with nanopipettes, they're reduced. So we can get a much better or clearer estimate of this ligand cofactor binding, the zinc, because carbonic anhydrase is a metalloenzyme and has zinc cofactors bound. So another thing to note with our nanopipettes is that the voltages that we use to generate the electrospray are generally much lower. So for standard emitters, we use around 1 to 1.4 kilovolts, whereas with nanopipettes, we generally use about 0.2 to 0.4 kilovolts to generate our electrospray. So we also wanted to see if we could test the sensitivity limits of nanopipettes. So we just sprayed myoglobin at 10 micromolar, 1 micromolar, zero point 0.1 and 0.01, so 10 nanomolar concentrations, and we can see that we can still detect protein charge states down to 10 nanomolar concentrations. And what's really nice with this is that the signal intensity kind of scales along with concentration. So I've mentioned that we use lower voltages, so that's really good for sensitive systems, especially proteins that have or biomolecules that have ligands bound or noncovalent complexes. We also found that we can spray, we can generate these very long, stable electrosprays with nanopipettes with limited clogging or blocking, which was to some of our surprise because if you imagine the pore is so small, the worry is that it's just going to block, but we don't generally see that. We're not 100% sure why that is right now. I have a theory that because quartz glass, I'm not a materials scientist, but quartz glass is very homogeneous compared to borosilicate. It could be that proteins adsorb to the surface less, and that reduces blocking, but we're not sure. But we do see these long, stable electrosprays at lower voltages. So next, we wanted to both increase the molecular weight of the analytes and also take it to a noncovalent assembly. So we analyzed malate dehydrogenase, which is a 70 kilodalton dimer, Ascend. And in order to get to spray with standard emitters,
Yeah, I think we lost Emily. Hopefully she'll come back.
did I just leave? I'm so sorry. My internet cut out.
Yes, you took a break.
I'm so sorry. This is so chaotic. I'm having so many technical
That's
difficulties.
okay. Come back in.
I'm so sorry. I don't know what's happening. I think I need to be admin again. I'm sorry. I didn't do anything. I was just sitting here.
No, I think your internet cut out.
I thought I'd come to campus to do it because there'd be less issues, but
Well,
I'm clearly
okay.
just not capable.
All right, start it up. Start it up.
Sorry. Sorry, everyone. I'm so glad this gets recorded so
Okay,
that
no,
everyone can see
okay, you're
that.
co-host. Go back to
Yeah.
malate dehydrogenase.
Yeah. Yes. It's super exciting stuff. My laptop just got overwhelmed. Okay. So yeah, we can see this also
Uh,
this asymmetric.
Wait. Let's see. You're still. It says you're co-host and you need to join Zoom again. Wait, I don't know. Hey, Paul, can she share her slides
I'm
now?
so sorry. Can you not see it?
Yes, now we can see it. Okay,
I'm
go ahead.
sorry. Okay. All
All
right.
right,
Sorry, everyone.
got it. Yep.
Clearly chaotic.
No, you're good.
Um, Thanks, Joe. Okay. Let me just check. Okay, we're good.
Yep.
Yeah, so you get this asymmetric charge patterning as well. So then we wanted to apply the nanopipettes, and because you get these finer droplets, you get better desolvation into the gas phase. So we can apply lower voltages at the source. So not only the in-source collision voltage down to 50 volts, but also the capillary voltage reduced from 1.4 to 4. And what we can see is like this really nice difference in the spectra. So we can isolate dimeric charge states that aren't overlapping with monomeric charge states. And we found that this system was also particularly sensitive to voltage. So if we reduce the capillary voltage even further down to our standard like 0.4 kilovolts, you can see the abundance of the dimer goes up relative to the monomer. So you can get a really nice isolation of this dimeric species and preservation of these noncovalent assemblies. And then what we can do is, because the intensity of that 16 plus is no longer overlapping with the monomeric charge state, we can isolate it and perform top-down fragmentation and get nice sequence coverage of that. Okay. Next, we then wanted to continue to increase the molecular weight of our analytes. So we applied our nanopipettes to GDH, or glutamate dehydrogenase, which is a hexamer around 335 kilodaltons. And it actually has a diameter of around 8 to 8.6 nanometers. And also beta-galactosidase, which is a tetramer around 460 kilodaltons and has a diameter of around 18 nanometers. And the reason that I'm telling you the diameters is because, remember, the tips that are making at UCLA are around 20 to 30 nanometers. So if you imagine, I'd like to imagine what we're seeing is sort of single ions coming out of the tip, which is generally what these nanopores are used for in, or nanopipettes are used for in nanopores and electrochemistry. So with both of these proteins, you can see that we're able to produce a native charge state distribution, and it recapitulates what's been seen in the literature. And another important thing is that we didn't really see any dissociation products, so we're able to preserve these oligomeric assemblies. And whilst this example here is only for 30 minutes, I was taking lots of acquisitions throughout, but we saw that these proteins were able to spray over long periods of time, even at 0.3 kilovolts that we were spraying at. So next we went even further and we tried GrowEL, which is around 800 kilodaltons as a tetradecamer. And you can see, again, still we're able to spray this protein at 800 kilodaltons through our nanopipettes, and also performed a concentration dependence, so 10 micromolar all the way down to 10 nanomolar. And you can see this charge state distribution stays really nice throughout and recapitulates what's been seen in the literature. So the examples that I've shown you since the amyloid ones have all been performed in ammonium acetate buffer, and I think that's great, but I think also the potential of the nanopipettes is to show that they spray things in different buffers. So an example of that here is BSA. So this is a collaboration with the Eisenberg lab at UCLA. Whilst I know you don't typically spray proteins in water, you can sometimes get a signal. BSA isn't very happy in water or ammonium acetate. By negative stain EM, you can see it forms these amorphous aggregates that deposit on the grid, and we can't see any charge states by native mass spec. Whereas in phosphate buffer, such as 5 millimolar, 15 and 30 millimolar, BSA is much happier. It doesn't form these amorphous aggregates, and you get sort of deposition of monomeric species on the grid. And what we can see by mass spec is that nanopipettes are able to spray BSA in these phosphate buffers. So this sort of expands the toolkit or the amenable analytes to native mass spec. If you think of lots of proteins or systems that are not happy in ammonium acetate, it could be now that we can keep them in their native buffers that they're happy in and get a nice native mass spectrum. And then the last example that I'm going to talk about is really pushing nanopipettes towards more complex matrices. So what we've done here is inspired by Mikhail Sharon's work, where they've done lovely analysis of proteins from bacterial lysates or lysate systems. So simply all we did here was either transform cells or bacterial cells with an empty vector that doesn't express a gene, so just a vehicle control, or co-transformed it with a plasmid that encodes an enzyme that acetylase enzyme, and also a plasmid that encodes α-synuclein. So we've shown in previous work that this system can produce recombinant α-synuclein that's 99% acetylated. So transform the cells, do an overnight culture, and then do little 50 mil grows and induce this system to express acetylated α-synuclein for 3 hours. Pellet the cells, wash the pellet once in ammonium acetate, sonicate them, and then pellet out the cell debris so that we're left with the cell lysate in solution. And remarkably, what we see when we then spray this lysate from nanopipettes is we get this really nice characteristic acetylated alpha-synuclein charge state distribution. It looks remarkably clean. And then obviously if we spray the system that doesn't express a gene, we don't see those alpha-synuclein peaks, but perhaps we can start to see some of these endogenous bacterial proteins down here. And then if we take this empty vector system and spike in some recombinant alpha-synuclein to that lysate, you can see that we can recover these charge states and see alpha-synuclein there. So this is kind of showing that nanopipettes can allow us to perform native mass spec from these complex matrices and can sort of extend native mass spec beyond purified preparations and systems. And then if you zoom in on the peaks that we actually see, you can see that they're really nicely desalted. So we've got the 10 plus and the 8 plus here. There's minimal adduction. And of course, perform this on a Synapt G2SI, so we can do ion mobility. And we can see that in the case of this recombinant lysate expressed alpha-synuclein, we get this drift time distribution, and that recapitulates what we've seen previously in the literature from recombinant proteins. So we're really able to capture these native proteoforms still from this system. So I hope, even with some issues, I'm sorry about that I've been able to show you that nanopipettes offer us to obtain native mass spectra at lower capillary voltage, so gentler source conditions, and produce spectra with fewer non-specific adduct peaks and a higher signal-to-noise ratio compared to standard borosilicate emitters. And we're able to preserve non-covalent interactions, so cofactors, we saw zinc still bound to carbonic anhydrase while we removed some of the non-specific adducts. And also we saw that protein assemblies, oligomeric states, were able to be maintained into the gas phase. And it also performed direct analysis from crude lysates. So I think this work really positions nanopipettes really nicely to analyze proteins and other biomolecules in systems that more closely represent their physiological environments. And we've also shown that they have cross-vendor applications. So data I've shown you here was performed on Synapt instruments and Orbitrap UHMR, Orbitrap Ascend. So this cross-vendor application means that I think they can be expanded more broadly across native MS. So things like direct mass technology, charge detection, top-down fragmentation, and ion mobility. So with that, I'd love to thank everyone that's been a part of this. So my postdoc supervisor at the University of Leeds, Dr. Anton Calabresi, our little nanopipettes team, so Professor Paolo Actis has been instrumental in this work, Dr. Chalmers Chow and Dr. Emma Norgate, also Professor Frank Sobot and Professor Sheena Radford at Leeds, and especially the British Mass Spectrometry Society for funding my holiday or like my research visit to Joe's lab for 3.5 months. And then also, of course, Joe and Rachel, the Lou Labs at UCLA for having me and helping get this work going. The Eisenberg Lab at UCLA and also Rafa at Thermo San Jose. And thank you all for listening. And I'm very sorry for the technical issues.
Good, thanks. Thanks, Emily. That was a great talk. Does anybody have any questions? You can either just raise your hand or type a question in the chat. Okay, so let me see. Let me just read. So Pei Su has a question here. Very nice talk. I wonder what is the typical flow rate, in these nanopipettes and the protein residence time in the tip region?
Yeah, that is a great question. I don't think I could give you numbers, but yeah, we did kind of look at that in some work. But it's I mean, it's just going to be much reduced compared to standard systems, but I
Well,
don't
so
have
how,
numbers.
okay, so yeah, Rachel's gonna, she's busy trying to calculate this. So
It's okay.
well, we wanted to
I'm
know
glad someone is.
also, we want to see what the droplet size is and all that stuff.
Yeah.
But how much material, what's the volume of liquid that you load in the back end of the tip?
So I think the max you can put in is 10 microliters. I usually do less. We did do we loaded 10 microliters and let it spray overnight, and then it literally sprayed until the entire solution was gone. I can't remember how many hours it was. I think it was around 17 hours. So you could probably work it out. We could probably work it out from that. But
Could you load 1 microliter
yeah, yeah,
and okay, so
and
that
let it,
would be a good experiment to do, would be to load 1 microliter and then
yeah.
just see when it just poops out. And then you can just
Yeah.
calculate the flow rate that way.
Just leave it, let it go, and then see when it stops. Yeah, that's good.
Yeah.
Do that.
Jenny Broadbelt's here, so she has a question.
Great. Thank you for the nice talk, Emily. I had a question. You mentioned, how long these can spray and so forth, but I think everyone who's tried these has had clogging problems. And so I'm wondering if there's any extra tips about how to avoid clogging or what you've done to make sure that yours are reliable and routine, especially with precious solutions that you might be loading.
Yeah, it's a great question. So I don't know how far I've seen quartz glass applied. I think a lot of the submicron emitters I've seen have been borosilica. So I think the difference here, like I say, I can't fully explain why I think it occurs, but it does seem to be that the quartz glass is better at enabling these longer sprays.
So that's the difference, Jenny. I think the people pulling borosilicate using the P1000 or P97 pullers, you get clogging, obviously. And so if you go to the P2000 and getting quartz tubes, then you'll have much better results. So I expect everybody now who's doing native mass spectrometry in these little in the regular glass emitters, I think they should switch over to quartz.
Me too.
Eric, you have a question.
I do. Hi, Emily.
Hey, Eric.
Good, nice talk. I
Thank
unfortunately
you.
could join the talk a little later, but just a follow-up comment from Professor Brockbelt. I was wondering about that too. I mean, in my experience, so micrometers they work fine for the standard conditions. For example, in ammonium acetate, I have to spray
Yeah.
up to 40 minutes, no problem. But when you have 150 millimolar sodium chloride, then that's not the case. That's what I wanted to ask you. Under what conditions you track the longevity of the spray?
Yeah, I guess most of the times where I've tracked how long they spray have been in ammonium acetate. Um, I mean, I've done a lot of analysis in the other buffers and I haven't really seen major issues, but as you say, I've not really left them for as long as I possibly could. That'll be the next thing to definitely do is like have PBS or really high salty conditions and see even the lysates as well and see how far we can leave those until they block. I assume they're probably less time than
Yeah,
just
yeah.
ammonium acetate.
No, you brought up a good point because some people, I mean, I always say, well, they tend to clog, but I always specify, well, they tend to clog under very salty conditions, and then ammonium acetate is not a problem. Now, my
Yeah.
question is, you in slide number 20, you show a spectrum where the charge distribution switch, right, for the monomer instead of an extended conformation, let's say that one here at the bottom left corner is switched
Yeah.
toward yes, toward relatively higher charges. I
Yeah.
remember Professor Williams. He reported surface-induced unfolding for cytochrome C. I don't remember exactly the system he was working with. I don't know if it has something to do here when you switch from just standard regular emitters or conventional emitters
I am
to
listening.
nano pipettes, if that's what you are observing here. Of course, it's gray, no? But I remember that paper from Professor Williams.
It's a really good point. I think what's occurring here, you're talking about losing these 6 plus and 5 plus. I'm not sure if it's that the collision voltage, when it dissociates the dimer, is you're getting like this asymmetric charge distribution. But I'm not sure if it's a surface effect.
But that's a good point, though. We
Yeah.
had not looked at that, and I remember Evan's study on surface-induced unfolding. That could happen. I mean, you have more surface coverage now with a narrower channel, and so
Yeah.
that could be something to look for later.
Yeah. Yeah.
Yeah.
Thank you.
That's it for me. Thank you very much, Emily.
Thank you. Thank you.
Okay, we have a question from a person that I cannot pronounce his name. Sorry.
Yeah. Thank you, Joe. Thank you, Emily, for your presentation.
Thank
Few
you.
years back in my lab, we have this P1000 puller. That's what we use, and we were pulling borosilicate glasses. And at some
Yeah.
point, we started coating with gold, and they kind of gave better results. So my question is, have you thought about coating these glasses with gold. Is it possible? Is this something that might maybe improve the kind of results you're getting already?
Yeah, it's a really good point. I don't coat the nanopipettes just because the coating, the pore is so tiny, like 20 nanometers. I'm worried that the coating is either going to block the pore or 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 manipulate it in some way. So I mean, I could try, but I'd be scared. But so that's why we typically put the platinum wire in the back to charge that way instead.
Thank you.
Yeah,
We,
thanks.
we should do that. I mean,
Let's just do it.
you're just scared, right? I don't understand. I mean, you said
I'm always scared.
you were scared to ship these tips to
Yeah,
Thermo, right? Right, and they were fine,
yeah.
right? So
Yeah,
so
I
we
put them in my suitcase back to Leeds and they were fine. So
yeah.
I mean the quartz glass is so strong. So yeah,
Yeah.
let's
Okay,
coat them.
here's another question on the chat. So, great presentation. I wonder if the nanopipettes work well for eliminating effects of glycerol. Oh, that glycerol
Ooh,
present in storage buffers of proteins. Oh, that's a good one. What do you
it is
think?
a good one. I haven't tried it, but it would definitely be really good if they could, because obviously so many proteins are stable in glycerol. I would still be scared to spray that. I think sometimes what we do when we're in salty buffers or really mass spec unfriendly buffers is offset the of course with Synapt sources the geometry is that the tip is offset anyway. But with thermo sources, I offset the left or the right a little bit to try and minimize, even though the flow is so low, but salt's going in. So perhaps with glycerol, that is something we could try. But yeah,
I like that one. That's a good one.
yeah, on your
That'll be the
instruments.
sure. When you come over in the summertime, let's just do this. Why not?
Let's just do all these. Yeah, okay, let's be brave.
Yeah. Anybody else have any crazy questions for us to try?
Yeah.
So I had a question actually. You started this project using or collaborating with that lab, Paolo's lab, who does nanopore sequencing.
Yeah.
What are the typical diameters for nanopore sequencing? Is it the 20 nanometer diameter tips?
They have a lot of protocols, and they change the diameter a lot. They can go up to I think 100 nanometers. But they also change the geometry of the tip as well. So the taper, like the length of the taper and things. We found that this protocol worked really nicely. We did try another geometry that had I think a longer taper. I might be wrong. I can't remember if it's shorter or longer. And they just didn't spray. Um, I tried to apply backing pressure and the tip just moved out of the holder.
Hmm.
Um, so they do use varied sizes. So we could sequentially do, try and do 10 nanometer, 20, 30, 40, and see what kind of effect. Because I think what we've kind of seen is that the tips made in Leeds are around 40 nanometers and the ones at UCLA were 20 to 30. And I
Yeah.
think the ones at UCLA worked much nicer and much better, or, clean things up a lot more. So it would be really nice to see like a sweep of sizes and how the desalting effect improves with tip diameter, because it might be that some analytes perhaps spray better from, um, a 50 nanometer pore, um,
Yeah.
but knowing how efficient it's going to be.
Yeah.
Yeah.
Yeah. Okay. Anybody else have any questions, suggestions?
Crazy
Uh,
experiments.
crazy solutions that Emily should try? Uh, okay, if not, then let's thank Emily again then for a wonderful talk. And I guess we'll end it here, Paul, and then we'll have another talk next month for the Proteoform Thursday. So thanks very much, everybody, for joining us, and we'll see you again next month.